Today's lecture focuses specifically on genetics. Genetics is the study of heredity and variation. Heredity really focuses on how traits are passed or transmitted from generation to generation, and the variation that we see associated with those traits that account for all the differences that we can see in appearance in individuals. In this example, what you see is two individuals who are reproduced to produce offspring. You can see the outward appearance of the two individuals, both having dark hair and dark eyes, and the outcome in this particular case is one child looks like you would expect with dark hair and dark eyes, yet the other child looks different in appearance than what you expect based off of the physical appearance of the parents. This child has blue eyes and blonde hair. Hopefully what you will get from the series of lectures that we're going to focus on is how, in fact, this type of situation can take place. Before we can continue on talking about genetics, what we need to do is review a few concepts. The first concept is a mechanism of reproduction. That is, some organisms have the ability to reproduce asexually. What that means is that they produce new individuals that are genetically identical to the original parent. There is no mixing of genetic information from two different sources in asexual reproduction. This is common in single-celled organisms. They reproduce by simple cell division, which is a mechanism of asexual reproduction, like you see on the left-hand side with the amoeba. However, there are multicellular organisms, both animal and plant, like you see on this slide, that have the ability to reproduce asexually as well. Sea stars, for example, can break off one of their arms, and one of those arms can develop into a new individual. Plants are very well-known examples of asexual reproduction, where runners or tubers or additional components can be produced to produce new organisms that are distinct from the original, yet genetically identical to the original. However, when we focus on genetics, we focus on a different mechanism or mode of reproduction known as sexual reproduction. In sexual reproduction, simply what we have in biological terms is the mixing of genetic material from two distinct gametes. In the slide, it's listed here as an example of fertilization of an egg by sperm. However, if you have two distinct gametes that come together, this can also result in sexual reproduction. The point behind sexual reproduction is that you have a mixing of genetic material that allows for a new genetically unique individual to be formed from this mechanism or mode of reproduction. And when we focus on genetics, the concept of sexual reproduction is something that needs to be clearly understood before we continue on with genetic principles. So where we start off talking about genetics is looking at the work of Gregor Mendel. He is considered the father of modern-day transmission genetics, that is, how genes or traits get passed on from generation to generation. It turns out at the time of Mendel's work, there were two hypotheses on how traits were transmitted. The first one is referred to as blending inheritance, and blending inheritance proposed that somehow genetic material was mixed, and eventually you got this mixture in the new individuals of the genetic material. However, with blending inheritance, the concept was that traits were blended together and they were not distinct in the offspring. So, for example, if you had a very tall parent and that individual mated with a very short parent, you would expect all of the individuals in the offspring to be sort of medium or intermediate height, as the two extremes in traits were blended together in blending inheritance. However, what Mendel had proposed in his work is something referred to as particulate inheritance. He proposed that parents passed on discrete heritable units that retained their identities in the offspring and were not blended or mixed together. We will see how he came to these conclusions in just a little bit. One of the other things I always like to mention is that Mendel was one of the very first individuals to analyze patterns of inheritance and document those statistically. It turns out that his work was actually uninterpreted or not well interpreted for a number of years, simply because he added the statistical analysis to it. Mendel's organism of choice was the garden pea. He worked on the garden pea because they were very easy to work with, they were easy to grow, there were many distinct or distinguishable varieties that were readily available with a clear history in ancestry. They can be very easily manipulated, meaning the female and the male components can be separated very easily so that anyone interested in doing so could form crosses that they wanted to do because the male and the female components were very easily identified and separated. It turns out that what he was interested in overall was trying to make new flowers or new flower colors. And from his body of work, again, is the foundations of transmission genetics that we know today. So what Mendel worked on, or the terms that Mendel utilized, was what he referred to as a character and a trait. A character, in Mendel's definition, was a heritable feature that can vary among individuals. The trait is the variant or the variation of a given character. So what you see in this table is an example of the characters and the traits associated with those characters. So for each, on the very far left column, you see the character name, seed shape, seed color, pod shape, and so on. And it turns out that there were two very easily identifiable traits associated with character. So if the character he was interested in was seed shape, it turned out that there were two possibilities for seed shape, round versus wrinkled. If he happened to be studying the character seed color, there were two possibilities there as well, yellow versus green. So again, there were characters and there were traits that Mendel was able to very easily describe and work with. Mendel performed a large variety of crosses. One example is what you see here in this slide. The first thing I want to do is refer to some definitions so that we can understand what exactly it was he did. This is referred to as a monohybrid cross, and simply what that means is that in a monohybrid cross, you look at one character and one character only. So in this particular example, he looked at plant height. Didn't care what the flower color was, what the seed shape was, what the seed color was. In a monohybrid cross, he focused only on one character and looked at only that character. So again, in this example, he looked at plant height. Some additional definitions associated with crosses is the parental generation, designated by the letter P, and this just indicates the individuals that are utilized in the initial cross. The offspring from that cross are referred to as the F1, which is short for first filial generation. The word filial comes from the Latin filius, which means son or offspring, and so you're looking at the first offspring in this cross. In most genetic crosses, the geneticists typically continue on by crossing individuals from the F1 to one another and looking at the next generation, which would then be referred to as the F2. So in this particular example, he worked with what was known as true breeding plants. That means that the plants in the parental cross came from a long line of plants that only ever produced that specific trait. So the character he was focusing on in this example was plant height, and the two traits were tall versus dwarf. So the tall plant in the parental cross came from a long line of plants that only ever produced tall plants. The dwarf plant in the parental cross also came from a very long line of plants that only ever produced dwarf plants. In this cross, he looked at the F1 and found that every single individual in the offspring were tall, and this caused him some concern. in that it seemed like the dwarf trait was somehow lost overall. However, he then took individuals from the F1, crossed them to one another, and looked at the offspring, which is known as the F2. In the F2, he saw plenty of plants that were tall, but he saw the reemergence of the dwarf trait, which seemingly was masked in the F1. Specifically, he actually counted up the number of individuals in this cross. There were a total of 1,064 plants in this cross. 787 of them were tall, and 277 were dwarf. What Mendel did was use a simple ratio calculation of the number of tall plants to dwarf plants. So if you take the number of tall plants, 787, and compare that to the number of dwarf plants, 277, you get a ratio of about 3. What that really means is that for every one dwarf plant, there were approximately 3 tall plants. This is a ratio of 3 to 1. Mendel didn't stop there. He performed a number of crosses with all of the characters that were in the table that we saw a few slides back. He repeated those experiments with monohybrid crosses, looking at all of those characters, and came to the same conclusion. Every single time, he found that there was a 3 to 1 ratio that resulted in the F2. What he was able to notice then was that there was a clear-cut statistical pattern associated with the characters and the traits that he saw. Utilizing that information, he proposed several hypotheses to help explain the results that he got. The first hypothesis that he proposed was that alternative versions of genes account for variation in inherited characters. We now know that these alternative versions of genes are referred to as alleles. So, with Mendel's terminology, the word gene that we use in modern day terminology is what we would refer to, or Mendel would refer to, as a character, and the traits were then alleles. So what he proposed again was that there were alternative versions of genes that allowed for the variation that he saw overall. Based off of those statistical patterns, he also hypothesized that for every single character that he analyzed, an organism will inherit 2 copies, one from each parent. Now, if you superimpose our understanding of what happens during meiosis, then you have a really good understanding of chromosome movement and what is predicted to be taking place by Mendel. Now, Mendel had no idea of what was going on with chromosome and chromosome movement during meiosis. He simply based this hypothesis based off of the observations that he saw. So this was truly revolutionary in that, again, we have a better foundational understanding of what's happening during meiosis and chromosome movement, and Mendel had no such understanding. He, again, based it all off of the work that he did and the statistical analysis that he performed. So just to reiterate, he proposed that for every character, an organism will inherit 2 copies, one from each parent. So again, for every character, every gene, there are 2 copies, one coming from each parent. In addition, Mendel also hypothesized that if there are in fact 2 copies of every single gene, if the alleles that you have or the copies that you have are different from one another, one is going to be referred to as dominant and the other recessive. The dominant one is going to be the one that we see the effects of in the individual, and the recessive one is going to be masked. When you have 2 copies that are the same, it's clear cut that the information, the genetic information that's present, will dictate what it is that we see in the individual. But again, if you have 2 different copies, one will be masked and one will be seen or expressed. Lastly, he proposed that the 2 alleles for each character segregate, or with our more modern day terminology, separate during gamete production. So again, Mendel had no understanding of what was happening during chromosome movement in meiosis, yet he proposed that if there are in fact 2 copies of every single character, that they must somehow separate during gamete formation. If you know the genetic makeup of individuals in a cross, you can then utilize that information to make predictions about the potential offspring. This example that we see here is utilizing what's known as a Punnett Square, and a Punnett Square is just a really easy way to do the bookkeeping to make the predictions associated with a genetic cross between individuals where you know the genetic information. So let's take one step back and define a few words before we move on to what's happening in this particular Punnett Square example. We're going to add in the terms genotype and phenotype. The term genotype refers to the genetic composition of the individual. What alleles are specifically present in the individual for a given character? That is the genotype. The phenotype is the physical appearance that we see due to the genotype. So, back to the concept of dominance and recessiveness, if an individual has 2 copies that are different, one will be dominant and one will be recessive, then the phenotype will be the dominant phenotype. In addition, when we talk about genotype, we can be more specific with respect to the genotype. A homozygous genotype is a genotype where the 2 alleles are exactly the same, and a heterozygous genotype is going to be one where the 2 alleles differ from one another. So, going back to this example, if we have a cross between 2 individuals, one individual being tall and the other being dwarf, again, in Mendel's case, these were true breeding plants, meaning they came from a long line of plants that only ever produced that particular trait. So that goes to say that the tall plant was homozygous for the tall trait, and the dwarf plant was homozygous for the dwarf trait. Mendel's prediction, or hypothesis, one of his hypotheses, was that during gamete formation, those 2 copies are going to separate from one another. So that's what you see here with gamete formation. What you can do then with a Punnett Square is list all of the possible gametes that would form from one individual along the side, and the possible gametes formed by the second individual along the top. Using the Punnett Square, you can then make predictions about fertilization events, where if the first gamete is fertilized by the first gamete in the dwarf plant, this would be the genetic combination that we would expect. And you can fill in this information continually, making predictions about fertilization events, which would then result in a specific genotype outcome in the offspring. So in this particular case, what we see is that all of the offspring, 4 out of 4, are predicted to be heterozygous, and all of the offspring, 4 out of 4, are predicted to be tall. So the genotypic outcome is 4 over 4, or 1, that they will all be heterozygous, and the phenotypic outcome, because of the classic dominant recessive relationship, is that all of these individuals will phenotypically be tall. In addition to the 4 hypotheses we just talked about that Mendel proposed based off of his monohybrid cross work, he also proposed another concept, which is known as the principle of independent assortment. And in this particular case, what he proposed was that if we are looking at 2 different characters, that when gamete formation occurs, they will assort or separate from one another independently of each other. And that's what you see here, again, overlaying our understanding of what happens during meiosis with Mendel's proposal of independent assortment. What Mendel did next was perform what's known as a dihybrid cross, and he performed several of them. A dihybrid cross is a genetic cross between 2 individuals looking at 2 characters and 2 characters only. So, for example, in this case, what Mendel did was look at peas as far as their shape as well as their color. So again, two distinct characters, pea shape and pea color. In this cross, what he did again was work with true breeding individuals. That means that they came from a long line of plants that only produced that particular character or trait. So he had plants that were round and yellow and plants that were green and wrinkled. Because they are true breeding plants, we can assign genotypes associated with those particular characters, and that the round yellow plants were homozygous for the color yellow and homozygous for the round shape, and the plants that were green and wrinkled were homozygous for the green color and homozygous for the wrinkled shape. When he looked at the offspring in the F1, he found that all of the offspring were yellow and round. What this suggests is that yellow color is dominant to green, and round shape is dominant to wrinkled shape. We can, however, utilize a Punnett Square to make a prediction about the offspring in the F1. Now, the Punnett Square is larger than the previous Punnett Square because we are looking at two different sets of characters rather than just one. In this case, we consider every possible combination of gametes that can be produced by the first parent, the yellow round parent. Because the only genetic information present in the yellow and round parent is yellow and round, we know that when gametes form, that's the only information that can be passed on to the next generation. We can hold that same logic through for the green wrinkled plants. Because they come from true breeding lines, the only genetic information around is for green and wrinkled peas. So the only genetic information that will be passed on to the next generation will also be the information to make green and wrinkled peas. So if you fill in the box or boxes in the Punnett Square, considering every possible fertilization event, what you should quickly see is that every individual in this cross that results will be heterozygous for color and heterozygous for shape as far as genotype is concerned. So if we take this cross to the next level, that is cross individuals from the F1 to one another, again, we know that they are heterozygous for both color and shape, and phenotypically they are all yellow and round because yellow and round are dominant. We can utilize a Punnett Square to make a prediction about the offspring. Now, in this case, the possible gametes that would form are going to be a little more complex than the last ones we saw, and that's because the genetic makeup of these individuals is more complex than those in the parental generation. In this case, you have to consider every possible combination of alleles that can come together during gamete formation. The best way to do this is to systematically consider every possible combination. The first dominant allele for color with the first dominant allele for shape. That goes in the first column. But you could also have the first dominant allele for color and the second recessive allele for shape. Or you can have the second recessive allele for color and the dominant for shape. Or lastly, the second recessive allele for color and the second recessive allele for shape. So this is every possible combination of color and shape alleles in gametes that can be produced in these individuals, and they are listed across the top. You would do the exact same thing for the second individual and list them all across the side. The next step is then to fill in all of the boxes with the predictions of genotypes that would result in the resulting offspring. What we see if you filled in the boxes would be that in the F2, 9 out of 16 would phenotypically be yellow and round, 3 out of 16 would be phenotypically yellow and wrinkled, 3 out of 16 would phenotypically be green and round, and 1 out of 16 would be phenotypically green and wrinkled. This is the classic 9:3 to 3:1 ratio, and this is what Mendel saw again for all of the dihybrid crosses that he performed.